by gathering samples in Copenhagen Zoo in Denmark and Hamilton Zoo Park in the United Kingdom. Two groups of researchers, including Dr. Elizabeth Clare and Christine Bowman, tested whether airborne DNA could be used to detect different animal species.
We can track DNA from various animals by filtrating the air, so we could track animals. It's firstly important in order to study different species. Secondly, we are in the midst of biodiversity crisis, and if we want to keep track of all different species out there and where they might be in decline, then we need efficient methods to do so to map their prevalence.
When living species leave waste, bleed, or shed skin or fur behind, they release genetic material known as environmental DNA or eDNA. Conservation biologists have recently sequenced waterborne eDNA to follow some species in aquatic habitats. Monitoring airborne eDNA, on the other hand, proved more difficult due to the fact that it is thinner in air than in water. According to UK study lead author Elizabeth Clare, the team working at the Hamilton Zoo Park was able to identify DNA from 25 different species of animals, including tigers, lemurs, and dingoes.
Actual experiment we conducted was to look at whether or not within a zoo environment where the animals are confined, we know where they are spatially, but also we know exactly what they are, whether we could recover that measure of biodiversity. So could we sample species in an area and then match what we found to what we know was supposed to be on site.
Despite the fact that the two research teams employed different ways to filter DNA from the air, both were effective in identifying the animals lurking close, both inside and outside the zoo.